line, apologies for having to listen to me, or congratulations, you haven't missed my talk, whatever the case may be. I'll be very careful, try not to touch too much. And if the slides stop, I'll just, damn the torpedoes, I'll just continue without slides, so we don't get any further behind in the program. So again, thank the organizers for giving us this opportunity to do our annual little update on what's going on at EBI in terms of archiving. And as always, also on behalf of Ardan, who unfortunately can't be here this year. And this is now the 4 people who are here as well in person with the photos. So if you have any questions or suggestions or comments on EMPIAR or EMDB, go and talk to them at the posters or chat with them on the Slack there, on Slack as well. So the 2 archives I'll talk about today are EMDB and EMPIAR. We also have PDB at EBI, of course, which archives the models, and the Bioimage Archive, which archives some 2D non-cryo EM data, and also the light microscopy, etc., parts of correlative experiments. But the bulk of the EM data archiving is in EMDB and EMPIAR. And as you can see from the statistics on the right, 2021 has been a great year for EM archiving. It's really going strong, even though the number of PDB entries as total per year is going down a little bit. EM is still growing strongly. So I'll update you a little bit about where we are with EMDB and EMPIAR, and then touch on some of the highlights of both these resources. And at the very end, I have a few words to say about the volume EM as well, that some of you may be interested in. So starting with the EMDB archive, damn the torpedoes. This is now part of the Worldwide Protein Data Bank. This is a collaboration between 5 different institutes that manage the 3 major archives in structural biology, the PDB, EMDB, and BMRB. And the growth, as you see, last year has continued to be very strong, even though 2020 was a bumpy year, probably because of the lockdown, etc. But still the numbers are up in 2021 again. And we're now, in fact, just a week away from the 20,000th released EMDB entry. So I think today we're at 19,949. So that will definitely happen next Wednesday. And it's quite amazing. 2 years ago, Ardan had a slide here to show that EMDB had to reach 10,000 entries. And now we're at 20,000 entries just 2 years later. And it's also this year's the 20th anniversary of EMDB. So if you look at the growth in terms of resolution, you see that after 2014, 2015, the numbers of EMDB maps at better than 4 angstrom resolution has really exploded. And it's even more drastic if you look at the percentage plot. So this shows for different resolution bands, the percentage of entries released in EMDB every year. And so in the past 2 years, more than 50% of all the entries that have been released have been at better than 4 angstrom resolution. So that's really quite impressive. In the archive itself and the deposition side, many things have happened behind the scenes and under the hood. And if you've deposited recently, you will have noticed some of the things. For example, that half maps are now mandatory if you use them for resolution estimation. And in the coming year, the team is working on proper handling of composite maps, which many of you have been asking for some time. And also we'll do better typing of auxiliary maps and masks, etc., and other files. And you'll be able to see all your maps and half maps and masks inside the deposition interface, which will hopefully reduce some of the mistakes that sometimes make it through to the archive. On the EMPIAR side, we recently had our 1,000th deposition submission, and that entry has also been released. If you look at the statistics, you notice that last year alone, we more than doubled the size of the archive. So there as well, the growth is really quite strong, both the number of entries and the size of the total archive. So we had 1 petabyte last year, and later this year we'll have our 2nd petabyte already. So again, the first petabyte took whatever it was, 8 years, and the 2nd petabyte will have taken just a bit more than 1 year. So part of this is because we get more depositions, but we also get bigger depositions. So if you only look at the cryo data sets, they have increased in average size from a couple of 100 gigabytes to more than 3 terabytes. And the biggest entries are over 40 terabytes and Well, the very biggest one is 75 terabytes. So I want to give these people a medal, metaphorical medal for having the patience to upload. That must have taken a month or 2 months to upload that kind of data to us. In terms of the holdings of EMPIAR, 90% of course is raw cryo data. That's what it was initially intended for, but we're also having about 10% volume EM and X-ray microscopy entries, FIB-SEM, serial block phase SEM, some correlative light in X-ray, correlative light in electron microscopy, soft X-ray and hard X-ray tomography. And also some data sets, this is a trend, are becoming more and more multimodal. So the one on the right is our most multimodal entry that we have with 9 different data sets from 7 different imaging modalities. And also, if you're watching this from Asia, to remind you that there's a mirror at PDBJ in Osaka in Japan, and they also provide a service if you have difficulties in transferring large amounts of data. They can help you with that. So you could send a disk to Osaka, and they will load it for you and help you with the submission. EMDB new, so there's 4 things. Only the first 2 of these I'm going to talk a little bit about. So one is the website, which was completely redesigned and launched last year in the summer. And another one is a project called EMIX, where we have collected cross-references and annotations from 20 different archives and resources in biology for EMDB entries, and then a little bit about validation as well. So the website was launched or relaunched last year, and I'd like to thank many of you who participated in our user survey about 2 years ago. We had over 100, I think about 120 people participated, and they weren't shy, so they told us what they liked, but also what they didn't like and what they missed. And I think almost all the suggestions and requests that we got have been integrated. So it's now easy to find half maps. You can query in the search system essentially every bit of metadata that has ever been supplied by anyone during a deposition. It's much easier to navigate, we think. So many improvements, and hopefully you've had a chance to work with the new website already. So the homepage looks fresh. The entry pages are very informative, rich in terms of their information. A very powerful search system. So I'll show a few examples of the EM stats with loads and loads of graphs and statistics. So if you're writing a grant or a paper, that's a good place to go to get the statistics on just about anything. An API if you're a power user, and then this integration of annotations and cross references to other archives. So the entry pages, as I mentioned, they are much richer. So almost all the metadata that we can collect and that people can provide if they are diligent is now also shown. The tabs make it easier to navigate if you're interested in the sample or the experiment or validation or etc. It's easy to get to. And if you're not a specialist, this is probably helpful as a biologist to be able to look at maps and tomograms in your browser without having to download any data or install any software. The EM stats, as I said, there's many different graphs now available, and all the graphs on the website are active. So if you click on any data point or a piece of a pie chart, it will launch a search of the search system and show you all the entries that fit that particular data point or slice of a pie chart. Also, the search system, you can make fairly complex queries nowadays, and once you have honed in on the set of entries that you're interested in, there's a statistics button here, which will give you plots and stats only pertaining to the result set of your search. So if you find all the entries on a particular molecule or from a particular institute or whatever, you can dig down and analyze these and produce nice graphs on the fly in the search system. Okay, EMIX I mentioned, so it means EMDB integration with complex structures and sequences and much more. So we now have collected cross-references and annotations from about 20 different resources, many from the PDB, from UniProt, from EuroPMC, which includes PubMed and PubMed Central, etc. And these are now available on the website through the entry pages, and also in the search system, and also in the API. And we'll soon make these also available via FTP, so other resources that expose EMDB entries can use this information as well. So just a few examples to show how it's integrated on the website. If you start typing something in the search box, like MEM here, you will get hits, text matching hits in the titles of entries, in the author names, etc. But also now in gene ontology terms, in PFAM assignments, PFAM domains, interpro matches, etc. So it makes it easy to find, for example, membrane-associated proteins, which we don't have annotated from PDB or EMDB specifically. There's also an advanced search option where there's a huge list of things that you can use to terms that you can use to search. And again, you can construct fairly complex queries interactively. So this, for example, selects all the entries that have a link to AlphaFold DB, but not to a fitted model in the PDB. So this gives you EMDB maps where there is no model in the PDB, but there is a model in AlphaFold DB. So if you want a quick structure paper, then you can take the AlphaFold DB model, fit it in the map, and submit your paper. And also, any search you do, there are these facets on the left-hand side that allow you to hone in on a particular set of entries. And at the bottom, there is this external links thing, so you can find all the entries that have, for example, a model in AlphaFold, or that have annotation in gene ontology or CADB or whatever. And on the entry pages, if you look in the sample tab, you will find links based on the UniProt IDs, for example, to UniProt and PDB-KB and AlphaFold DB. You'll find mappings on the sequence from Interpro, links to gene ontology terms. And again, if you click on this, it will launch a search and show you all the other entries that have the same gene ontology assignment. And then 2 slides about validation, but I'm not going to talk about them because they're both available for both use and for reading. So we have a fairly extensive validation analysis resource, which took input from the last workshop we organized before lockdown. So this was January 2 years ago at EBI. And many of the recommendations have already been implemented. And so this is now available. There's also a paper in last year's CCP-EM proceedings that came out this month in Acta Christi. So you can go and read all about it. And we're also working with CCP-EM to integrate the code into CCP-EM. So it will be at your fingertips in the lab as well soon. And another thing we talked about in past years is TrueDAW, which is a validation method that uses libraries of what sidechains look like at different resolution ranges in EM maps, and compares the sidechain features in your map to them to score them and to find if there's any places where you might want to have a closer look. This is now in CCP-EM as of version 1.6, so you can use this, and the results are you can view in Camera with a plugin. It's really nice and easy to use, and there's a preprint that you can access through the website, so you can read all about it as well. Okay, EMPIAR. On the EMPIAR side, again, well, lots of improvements and small changes on the website. So we've made some tutorials and collected some talks that are easy to access now. You can do searches of EMPIAR using the EMDB search system. So there's a link on the EMPIAR homepage, and also a tutorial which quickly explains how this works. So you can do slightly better searches than just using the table on the EMPIAR homepage. We have cross references, but only to EMDB and Bioimage Archive. So if you select EMPIAR entries that have a cross-link to the Bioimage Archive, it means it's a correlative data set where we have the EM or the XM data, and Bioimage Archive will have the light microscopy data. We also realize that we have hundreds and hundreds and hundreds of 3D volumes in EMPIAR. So we make those available now in this interface as well. So again, if you're a biologist, you don't have to download any data or any software. You can view these data sets inside your browser quickly. And something we will be announcing soon is the option to claim EMPIAR entries to your ORCID profile. So that means you can get proper credit for all the hard work and all the data sets that you've collected. And Gerard was our guinea pig, so he's already done this. It's a very simple procedure that very quickly allows you to add all your EMPIAR entries to your ORCID record. Okay, finally, I want to spend a few slides on volume EM. And some of you, I saw Michelle was here, some of you know quite well what this is, and others may be interested. So to paraphrase an old Prussian, I don't remember, it was Bismarck or von Clausewitz, but volume EM is the continuation of cryo-ET by other means. So this is the sort of the regime where cryotomography can be used, but then volume EM is here. So using FIB-SEM, SBF-SEM, array tomography, grid tape, TEM, etc., and other techniques, you can study with non-cryo methods the 3D structure of cells and tissues and organs and small organisms. It's very popular in connectomics work on fly brain, mouse brain, etc. And it's often used correlatively. So you use another technique to pinpoint regions where you may want to do your EM analysis. In the UK, we started a couple of years ago with the idea that we wanted to convince the funders to invest in infrastructure for this and get state-of-the-art instruments for everyone to use in the UK. So they started with a small meeting hosted by the Wellcome Trust with about 60 people from mostly the UK, but also a couple from the US and Germany, etc. This then began to snowball. We prepared a roadmap, we had a survey. The group grew and grew, and we had another town hall. And then the community got very enthusiastic and decided to self-organize and into a number of working groups to discuss and do things in the area of sample prep and outreach and training, etc., and data. And the third community town hall last year already had 200 participants from all over the world. And this year we are working hard to the working groups are doing their things, and we're trying to get some funding because all of this is just voluntary work. We're writing reviews and comments and news items and etc. And next year there will actually be a first Gordon conference in California on volume EM. And volume EM will also be, we started a new Wellcome Trust grant last year. So in this last year, in the coming 4 years, implementing proper support for volume EM will be a main focus of our work on EMPIAR. We had a workshop in December where we discussed things like data models, data dissemination methods and formats, because the volumes can be very big. So we need different ways of giving people access to using modern formats to those data sets interactively rather than forcing them to download terabytes of data first. And also on segmentations. So we want to start archiving segmentations in EMPIAR as well. So if you're interested in this and sort of getting on the ground floor, it's not quite the ground floor, but maybe the first floor, have a look at this website. And Martin Wynn is also heavily involved. Michelle Darrow is here as well, also heavily involved. So come and talk to us if you're interested in any of that. Okay, and I'll just give you the slide. So if you Google volume EM Gordon conference, you'll find this page on their website, I'm sure. Okay, so all I want to do next is to thank the funders, the Wellcome, MRC and BBSRC, who have funded the work on EMPIAR and EMDB, and also EMBI, EMBL, of course, for many years. And again, so Simone, Je, Nelly, and Jack are here in the meeting, and they have posters. So feel free and come and talk to them. And I think I haven't added too much to the running time of your session, Neil. So thank you for your attention. I'm happy to take questions, or we can do it in the Slack as well, but, whatever you want.